Good afternoon, everyone. Let's go ahead and get started. I know people are still coming in for their food. Dr. Peter Morgaine, I think everybody at least has heard the name before in various contexts. Certainly our Morgaine Fellows. If you look across the street, you've got Morgaine Hall. Dr. Morgaine was a colleague for many years. Worked with myself and others on the effects of prenatal protein malnutrition, but he also did a lot of work on cetacean brain. And after he passed away, I was given all of his archives, his paper archives, and a lot of the artwork and other pieces of research on the cetacean brain. At around the same time when he started to retire, he also had thousands of slides, histological specimens of whale brain. And that was first given to a colleague at New York City College, Ilya Glazer, and then that was transferred when Ilya retired to Dr. Patrick Hoff, our speaker for today. So that's a little bit of that. And over the past year or so, Dr. Hoff and I have started to work together to put together the Morgaine archives between his histological sections and the other papers that I have in the collection here at UNE. So Dr. Hoff has been working with me and others to go down. We went down this summer to New York and looked at the histology collection and started to work on that. And then he came up for a couple of days now to take a look at the collection that we have up here. And we want to meld those two together so that we have a valuable or invaluable resource for research on cetacean brains. Dr. Hoff graduated from the University of Geneva, got his MD there. He is the Irving and Dorothy Regenstreif Research Professor of Neuroscience and the Vice Chair of Fishberg Department of Neuroscience at the Icahn School of Medicine at Mount Sinai in New York City. He's also editor-in-chief of the premier neuroscience journal, the first, going back to the 1880s. The Journal of Comparative Neurology. And he's published extensively in a lot of different areas, but his real passion is in comparative anatomy, and he's going to talk to us today about the comparative anatomy of the cerebral cortex. So thank you very much for coming up, Patrick.
Okay, thank you. It's a great pleasure to finally to visit the University of North England and be on the grounds where Peter worked a lot. So I'm going to tell you the part of my story on the comparative anatomy of the cerebral cortex and a little bit about the relationship of that type of work with the human biomedical neurological field as well, and see how what you can learn from a species may apply to our own species. And through my talk, you'll see that we're working on a number of strange animals. You'll hear about the elephant and the hippopotamus, and evidently a lot about whales. So of course, you've got to get brains, and you wish it were as easy as just drive in, right? That thing is real. It exists somewhere in the Midwest. Forget where. But it frequently starts with making the right connections with people who do things in a little bit of an uncommon manner. And two of the people who were instrumental in my career in fostering my interest in comparative brain anatomy really were Peter Morgaine and this guy in the black and white photograph, Ilya Glazer. So I met them in 1990 at the Society for Neuroscience meeting, and essentially I went to them And I say, well, you know, what you do is really cool. I'd like to study the whale brain. And so how do I do that? And Ilya Glaser said, well, you are in Manhattan. I am in Manhattan. You cross town. You come to my lab, and we talk. And you know, Ilya was a completely no-nonsense person. So that picture of Ilya is interesting for historical reasons. It's taken from a little township near Krasnoyarsk in Siberia. Ilya was then in political exile in the 1960s due to unwanted political agitation, talking a little bit too loud about the cause of Russian Jews, and that was unpalatable for the then government of the USSR. So he spent 3 years in a gulag and then 3 years in political exile. After that, he escaped to Israel, and from there he became a professor at the City University of New York, where he taught anatomy and histology. And that's where I met him. So Ilya Glaser was the co-author of that famous book, The Human Brain in Tables and Numbers, that he had edited with his thesis mentor, Shmuel Blinkov. And so he was a real personality in the older times of neuroscience. I'm very glad to have met him. So with the help of these guys, we started to build up a collection of various samples. In here, you have brains from great apes. The brains are not at scale. So if you look at the brain of the fin whale, which is this one here, it's in fact a lot bigger, and you're talking of a specimen that is about that big. And we started to study cortical organization in these specimens. So when you look at let's concentrate on the cerebral cortex to start. When you look at the cerebral cortex across mammals, you will have sort of a generic blueprint with a lot of commonalities, and you can recognize a visual field and auditory fields, somatosensory motor fields that have a distribution that, relative to each other, is very, very highly conserved across the cortical mental, across all mammals. And that stems truly from a common ancestor, and you observe it in monotremes as much as you observe it in primates. So the only differences among species really is in the size of these different fields, how these different fields are organized based on species-specific adaptation to ecological niches and so on and so forth, and also to the size of the white stuff around these primary fields, which is evidently the association neocortex, where a lot of what we call cognition and emotion and higher behaviors are generated. Okay, so how does the cortical field scales up and change among species? So if we look at sensory domains like visual, somatosensory, auditory, these primary sensory functions, they will have a different representation as cortical fields from which you can record functionally with electrodes, right? And if you take a squirrel, for example, it's a highly visual animals. They see color very well among rodents. They have a big V1, but the auditory cortex will be looking diminutive, relatively speaking. They have also a fairly large somatosensory cortex. If you look at the platypus, everything is about the duckbill. And then they have a gigantic primary somatosensory cortex, not so much for their body, but just for the electrosensory organs of the duckbill, right? So there are modifications to the neocortex. It can be differences simply in the cortical sheet. You have very small species, very large species. So of course, that's going to give you more or less real estate to put function in there. It can be changes in the relative size of neocortical field, as we just saw. It can be changes in the magnification of behaviorally relevant sensory entities inside this representation. For example, the hand representation in primates, but in some rodents, the representation of the mystacial vibrissae. The addition of cortical modules in the field. So for example, auditory specializations in bats or the vibrissae in the rat and mouse. The changes in the number of cortical fields and the changes in the connectivity among cortical fields. So for example, if you look at the human visual systems, we are intensely visual, and we have an extremely sophisticated set of visual areas and connectivities that permits us to analyze our extrapersonal space, right? Which a rat doesn't do as much. They do something quite similar, but it's comparatively much more reduced. So these are some generic context to keep in mind. That's a representation of flat mount of the vibrissae in the rat. That's from the Weller collection. Different rodents have different type of barrels. In some species, like the muskrat, it's not particularly prominent. It still exists. Other species don't have barrels at all. Some species have barrels and whisk. Some species have barrels but do not whisk, and so on and so forth. And all of this is represented in the internal complexity of the primary somatosensory barrel field domain in these animals. This is the classical work of Woolsey. Other species do things different. They whisk with other organs. And for example, if you take the star-nosed mole, which is an animal that is about that big and has a brain that fits in a quarter, it has a remarkable specialization of its rhinarium. So there are fleshy appendages that really looks like fingers that comes out of the area of the nose around his nostrils, right? And they are fossorial. They live in the mud. You have one example of that beast around here on the top right panel, and you see the fleshy appendages of the rhinarium. And with that, they feel their environment. So, something like the platypus, right? Except that it's not electroreceptors. These are also a modified Eimer's organ, and they can feel the world and detect the type of worms that they prefer eating. And in their primary somatosensory cortex, you have a topographic representation of each of these fingers that come out of the nose, and that's really how they see their world. The other thing that is represented nearby that is that big, large fossorial hand. These appendages also have the capacity to manipulate stuff in their environment and literally make something similar to what we do with our fingers or what the walrus does with its jowls. It's called auripulation. That's the platypus. The platypus wants underwater when it hunts for crayfishes, does not rely on visions at all. They close their eyes, they shut down the lids, but they use exclusively their electrosensory sense. And that's reflected, as I said, in that major somatosensory representation of the bill, which is close by to the motor representation here of the duck bill. And that in contrast to what you see in macaque monkeys or flying fox. The flying fox will have a very delicate representation of the wing, similar to what we have with fingers and so on and so forth. So all of this results in differences in the cellular organization of these cortical regions, and within them cortical modules that are reflected in cell types, cell densities, columnar organization, resulting in widely differing cytoarchitecture among species, even if you stay within the same order. In that case, you can compare the same frontal field in a long-tailed macaque and chimpanzee and a human, and you can see obvious differences. Some of them just due to the fact that the human has a larger cortex and the apparent cellular density is actually less as the cortex expands compared to a smaller brain species. And at the local level within the same area, on the left side, you have the primary motor cortex in a series of primates. On the right side, it's the primary visual cortex in the same series of primates. The first one at the top here is a prosimian. It's a mouse lemur. The last one here at the bottom is the human. So in the human motor cortex and in the other species, you see the prominence of these BET cells, which are the large motor neuron, corticomotor neuronal tract. In the primary visual cortex, you see the presence of these minor cells that are important for motion detection and motion analysis in the periphery of the visual field. And you see that you have differences in size that generally scale with the size of the animal, but not entirely. And in fact, if you look at this guy here, in that species, you have these absolutely gigantic BET cells that turn out to be larger than the BET cells of the human or of other great apes. So that monkey here is called the patas monkey. And the patas monkey is the fastest running species of monkey on Earth. They are one of the best prey for jaguars, and they have to outrun. And they can beat them on a short distance. But they have this extraordinary development of the primary motor cortex. The other thing is that they have more minor cells per millimeter square in the primary visual cortex, because all of their survival depends completely on detection of the predator in a savanna environment, right? So everything looks pretty much the same, and they rely on vision as much as they may rely on smell. They also have interesting community behaviors with specific shrieks depending on what's in the neighborhood. And if there is a predator, you will see an entire troop start to run at once. They are fascinating animals. So you have this species difference within something that is fairly stable as a cortical system within the same order. And then the next step, once you look at purely cytoarchitecture, is to look at differences among cell types. So to do that, you have to go beyond the Nissl stain, look at specific markers. So using immunohistochemistry, you can find markers that will selectively label different cell type. Here, using simply neurofilament protein, we label interesting subclasses of pyramidal cells that, just looking at visual areas in a macaque monkey, show clearly regionally specific patterns in laminar distributions. And these large pyramidal cells are all responsible for furnishing cortical projection systems. So that's a cell type which distribution differs among functionally different cortical fields and furnish different type of cortical association function. And they are very useful because just using that simple stain, you can develop a map of the visual cortical system in a macaque, and you can do that in other species as well. For example, here, the exact same idea in a mouse brain, and you can see that you have this, on a much smaller scale, also a regionally specific distribution of this protein in specific cell type from the primary visual cortex to the secondary visual areas and tertiary visual areas in the mouse visual cortex. So again, that's commonality of organization, but with different densities and different laminar organization, different numbers of relevant cortical areas or modules across mammalian species. And that can be extended further. So if we look beyond the human and the classical lab animals, we got interested in things like this. This is the primary motor cortex of a fur seal, and it has these enormous cells here in layer 5. Bets-like cells, probably associated with swimming functions. And you found the same type of cells in the cortex of whales. Here you have a pilot whale, and this one here is a mysticete that was a humpback whale. Same thing, you can study now not excitatory neurons, but GABAergic inhibitory interneurons. So there you have a much broader registry of stain that you can use with calcium-binding proteins or neuropeptides that can subclassify within cell types different functionally types of cells based on their relationship. And if you compare a primate here to a beluga whale here, you can see that with the same marker, this is parvalbumin, so you're thinking of basket cells, for example, you have a completely different types of cells in the cetacean. So there you have something that has diverged during evolution with completely different patterns. But then you have other commonalities. On that panel below here, that cells is in layer 6 of the giraffe neocortex. It's a large calretinine positive neuron. It's a deep layer 6 inhibitory cell, probably Martinotti type, not sure exactly. And if you look here in a whale, you have images that are not incompatible with that, which would make sense because cetaceans are very closely related to terrestrial artiodactyls, and in fact are within an order that now is referred to as cetartiodactyls. And you can look at that and try to see where are the commonalities and where are the divergences. Here we use 3 calcium-binding proteins, parvalbumin, calbindin, calretinine, across animals that represents broadly diverging mammalian orders, including some a prototherian here and a marsupial, and here a megachiropteran, some primate, and a dolphin, and that you see that you have cell types that occur only in some clades, whereas other have some more commonalities here, which permits them to regroup them at the supraordinal level. So for example, here we have Archontogliers fairly nicely represented together with parvalbumin, calbindin, and to some extent calretinine, but note that in the cerebral cortex of the flying fox, there is absolutely no calretinine positive interneurons. Okay, and no, it's not that the immunostain didn't work, because calretinin is present where it's supposed to be present, in the thalamus of the flying fox. So they have calretinin, just calretinin in the cortex disappeared in that particular branch of mammals for whatever reason. And you can organize these staining patterns within a cladogram and see how it worked. So we had a study here of a particular group of mammals that now are regrouped to a super level called Atlantogenata, but essentially regroup elephant shrews and sloths, anteaters, and things like this, as well as the rock hyrax. The rock hyrax itself being closely related to elephants and manatees. So these are Afrotherians, and they have a pattern of interneurons that is quite common among them, but very different from what you find in cetartiodactyls or in carnivores and in archontoglyphs, where they differ also by their neurofilament positive neurons in terms of the shape of the cells that are labeled. They are excitatory neurons, but their morphology is very different than what you find in rodents, primates, and carnivores, for example, and so on and so forth. So you can start to see over a more mammalian generic taxonomic organization, things have evolved in certain branches where you have convergence, where you have divergence. What may represent an ancestral plesiomorphic state, what might be a synapomorphy or an apomorphy in distal branches. And then you can go beyond just protein localization and look at genes. So that's a little bit more difficult to do, because to do that type of work, you have to have materials that are tractable to in situ hybridization and preparation and preserve these materials. So this is in situ that I did in collaboration with the Allen Institute. They have their entire human atlas and mouse atlas with expression of all of the genes that are expressed in the brains of these creatures, right? So you can see things that have some commonalities. So this is just a temporal cortex in the human and the mouse, and you can see that these genes have a pattern of distribution in cell types and layers that is generally quite similar in the mouse and in the human. You know, for example, here you have cells in layer 3 and layer 5, layer 3 and layer 5, 6, so on. These genes not particularly expressed, just a little bit in the human, not at all in the mouse. But it's not totally true. And the NPY is very, very poorly expressed in the human brain. In the human cortex, it's much more expressed in the mouse, and so on. But then you have patterns that are similar between the two species in terms of laminar expression. So, for example, here you have genes that are expressed only in layer 1, some in layer 2, upper 3, with a little bit in the deeper layer, some more generally through the cortex, and some only in the deeper layers. And these patterns are conserved quite exactly for the two species. And then you have genes that are just expressed broadly through the cortex among different cell types with no particular organization, in a way that is also conserved among the species. So using that, you can see how a mouse and a human diverge in terms of the neurochemical phenotype of particular cell types. And that's very important to know in view of the use we made of mouse models for the study of brain disorders, right? And when you start to dig into that, you realize that really a mouse is not a small human. And that these models are really cool to learn a lot about the molecular biology of protein effector systems, but in terms of really anatomical function, there might be a lot more limited. So to take a case of an extreme divergence among mammals, cetaceans are a very good case study. So these brains all come from Peter Morgaine and Ilya Glaser. So you'll see that among cetaceans, there is a large difference in how the brain look, how much they are convoluted. First thing that they are very convoluted. They are a lot more convoluted than in any other species. So they have a greater cortical surface altogether, but it doesn't mean that they have that much more cortical volume because in fact the cortex is thin comparatively to primates. And there are other ways by which these brains differ. I just show the MRI here. As much as we can, we are documenting these brains as MR data set because that's a permanent record. Once you start cutting and slicing, it's destructive, so you don't have the brain after. And of course, the first thing you have to do is to get the brain. So it takes some work and some muscles too. Yes, that's where the graduate students and the technicians are happily involved. So you get, that's a minke whale. So that's a relatively small rorqual. So it's still manageable. You still have to have a crane capable of lifting the animal. So that was a 10-ton power crane. That animal was about 3.5 tons. And then you start dissecting. You separate the head from the rest of the body, and you use your saw, and you get to the brain. This last picture here is a view of the pharynx and vox box of the whale. I'm collaborating with an expert on the larynx evolution at Mount Sinai, and we expose here the larynx. You see that big nerve here on the side? It's about the size of my thumb. That's the facial nerve. It gets sometimes more exotic where you really need a lot. This is a humpback whale that beached in the Hamptons on Long Island. So nobody in the Hamptons wants a beached whale on the private beach, right? So the local sheriff gets here and says, okay, you're gonna have to get that out of that beach pretty quick, guys, right? But then that gets under control when the NOA shows up and the wildlife and fisheries and the local shaman. There was the local shaman who had to do the rites for the deceased whale, and after that we could start dissecting. So you have to have heavy equipment to carry the thing out of the surf, far away from the tide line. It takes a long time to go to it, and then you hack at it. That's a view of the heart. This was still a fairly juvenile specimen. They get bigger. And again, you know, we get the brain. You'll notice the rain gear. All of this was done under beating rain. And here is my colleague, Bridget, who is here, who was the only person with small hands enough to fit into that hole to get under the brain and lift that brain. So imagine the brain is that big, right? So, and here is the brain in my lab after it was removed. So we got the brain in superb condition, and that's a section not from that brain. That's a section that comes from the humpback brain that was in the Peter Morgaine collection. So this is a parasagittal section of the humpback whale brain, and you can see that extremely folded cortex in the hemisphere here. And here you have the thalamus, you have the basal ganglia, brainstem down here, and you can see also the extremely large cerebellum. With the elephants, these are the species with proportionally the largest cerebellum on Earth. So what's a whale? I said that the whales were apparented to artiodactyls, so to terrestrial animals. So the question is, what is an artiodactyl? So artiodactyls came out of condylarthra fossil species and Mesonychians, and they are all characterized by one bone characteristic, which is a talus bone in the ankle that has a double pulley. So there is an upper pulley and a lower pulley. If you don't have that, you are not an artiodactyl. So for example, species like this guy up here, this fossil up here, is still not quite an artiodactyl. Had a talus looking like this with only the upper pulley. But this guy here, the second fossil, if you take, you notice that it has hind legs, right? They were semi-aquatic at the time. It is a whale because Pachycetus talus has an upper and a lower pulley. And then progressively, they became increasingly aquatic as this species, by the way, Pachycetus means Pakistan, so you find them in the Indo-Pakistan areas. That's where the earliest whale fossils were discovered. And they were in an environment whereby, if you go to that lower level map, if you can discern here, in the northern part of what was going to be in Indo-Pakistan before the Indian subcontinent actually had merged with the Asian plate. So in between, you had the Tethys Sea, and from the early Eocene, where the first whale-like creatures evolved there, the Indian subcontinent closed the Tethys Sea, generated the Himalayas, and from there, you had a radiation through the waterways of cetaceans on the entire planet. And progressively, as that happened, they became, you can see the artistic rendering here, they became less and less able to ambulate on the ground, lost the hind limb, and eventually became entirely aquatic. In fact, if you take the skeleton of a modern balenid, like a bowhead whale, you will find a vestigial pelvis in there. In the baleen whales, in the mysticetes, not in the odontocetes. And interestingly, the mysticetes were the first to evolve, and the odontocetes are more recent. Okay, so that's for a little bit of paleontology. I promise you I won't bore you more with paleontology. And so now study the brain of the modern whale, and we compare it to a human here. So that's the primary visual cortex, with this very large layer 4. Everybody knows that well, typical of primate, right? That's the primary visual cortex of a dolphin, and well, there is no layer 4 in there. And in fact, the entire cortical mantle of cetaceans is characterized by a total absence of layer 4. Okay. That's a problem in context of connectivity, right? Because layer 4 is an area where a major input from the thalamus reaches the cortex, and from there into layer 3. So how does that happen in the cetaceans? Cetaceans clearly do not have spiny stellate cells in layer 4. So you'll notice that there is a gigantic layer 1 that occupies about a third of the cortical thickness. And that layer 1 is a lot more cellular in terms of neurons than what you find in terrestrial species. And we suspect that all the thalamocortical innervation, or nearly all of it, reaches up to layer 1, make contacts with interneurons located in layer 1s, and then goes back to the more cellular layers of the cerebral cortex. Other differences that you can see is sort of that columnar arrangement of neurons in layer 5 and 6, sort of these modules. And that's prominent in the primary auditory cortex of whales, in the primary visual cortex of whales. I'll show you that across species, very different from what you have in primates and rodents. So clearly, cetaceans are doing things differently. And among cetaceans, you have among different species differences in cell densities, while across cortical areas, certain general themes are conserved. So if you look into there with enough details, you're going to see patterns emerge, and you realize that in that cortical organization, you probably have as many cortical areas than you can find in a human brain. And then you want to look also at animals that, like cetaceans, adopted a more aquatic lifestyle. So one that makes a lot of sense here at top right, if you see my little arrow, is the hippopotamus. And the hippopotamus is very important because from the molecular phylogenetic point of view, it is the closest neighbor to all cetaceans. Okay? Hippopotamus are remarkably amphibians. I mean, they spend 90% of their time in water. There are commonalities, and we'll get back to that. We also look at seals, and this is a walrus here. And the seals and walrus have a cortex that looks much more like what you find in a carnivore, in a bear, or dog, or and that makes sense. Okay, so there are divergences there too. We compared it with that very strange brain here. This comes from a manatee. So, yeah, also a totally aquatic mammal, right? And manatee is essentially lissencephalic, except for a few throws in the cortex. There is a sylvian cleft here, and not much more. So why did that happen that way for an animal that is closely related to elephants? Who knows? But certainly an adaptation to very specific environments. And the cortex of the manatee is itself completely different from what you see in other mammalian species. So we compared different whale species. So you have these elements in the deep layers, these modules that are very visible. That's the primary visual cortex. Same thing in the primary auditory cortex. So maybe this in the auditory cortex could correspond to isofrequency domain coding in the auditory cortex. We have no idea of these functionalities, evidently. And then you have idiosyncrasies like that you observe. That's a humpback whale. These nests of cells in layer 2. So if you've looked at the entorhinal cortex in the human brain, you have these islands in layer 2 of the entorhinal cortex. Now, in the humpback whale, it exists on a huge domain in the occipital pole. That's not the entorhinal cortex at all. We have no idea what it does, but there is clearly these large columnar domains that exist under one of these layer 2 islands on a very large domain of cortex. So what it does for the whale in terms of cortical integration and integration of what modality, we have no idea. And then you have that. So look at these cells, these sort of spindle-shaped cells. And this is in the brain of the humpback whale. It's absolutely fascinating. These cells exist also in other cetaceans. That's a fin whale. Here you see them in the sperm whale very well. Here in a killer whale, you have example. And of course, we find them also in the human brain. So this is called a von Economo neuron. And that's where we're going to go back a little bit to the human brain. So Constantin von Economo was a Romanian neurologist. He is the person who studied the flu-related encephalopathy in the 1918, 1920, and described the lethargic encephalopathy related to the influenza pandemic in these days. But like Brodmann, he also issued a broad map of the cortical cytoarchitecture of the human brain in a map that is comparable, a little bit more detailed than that of Brodmann, actually. And he did not discover them, but he described them for the first time in great details, these particular spindle-shaped neurons, which now bear his name. And interestingly, we rediscovered these cells. One day, we were looking at cingulate cortex of human in my lab, and Esther Nimchinsky, a graduate student in my lab, said, come look at that. And we thought first it was an artifact of preparation, and she was seeing these spindles. And then looking at more cases, it was clear it was not an artifact. So for 3 days, we had the Nimchinsky cell, because we thought that we had found a new cell. And then we did our homework, and we looked at the old guys. So first report of it was by Betts in a series of papers on the fine structure of the brain of human in the Zentralblatt für medizinische Wissenschaften in 1881. Then a few other people, including Ramon y Cajal, cite them, and then von Economo and Koskinas describe them, particularly in that paper in 1926. Maximilian Rose described it in different species as well in his work. And then it disappeared from interest for many years until we stumbled on them. We stained them with neurofilament protein. So remember the neurofilament protein, very cool to look at excitatory neurons. We also reported the fact that we know they project out of the cortex. We put dye crystals, and we could trace a few of these cells. So they send an axon in the cingulum bundle, so they are projection neurons. So based on that, and their presence in the frontoinsular cortex, I had a great insight, and I said, okay, this is the upper motor neuron of the autonomic nervous system. Now we know. Yes, except that this was known since 1986, because if you read the last sentence of von Economo paper, you see here that most likely these cells are in a position to be efferenten sympathischen Funktionen. So he had the insight. And the reason he had the insight is that he had studied a paper of a neurological case that had a lesion of the anterior cingulate cortex and had urine incontinence. So it had to go through the sympathetic system and the control of the autonomic control. So just with that, he said, okay, this is great. We have the sympathetic upper motor neuron. And that's probably exactly what these cells are doing. There is another cell type in this exact same region that Economo, for some reason, did for sure saw, but did not describe, is these fork cells. And these were described by this Chinese gentleman who was working in the lab of Oscar and Cecil Vogt. And they adopt the same distribution in layer 5 of the anterior cingulate cortex, but more prominently, the frontoinsular cortex. We don't know their function. Interestingly, so that's the distribution of these cells. You find them really in the anterior portion here of the insula and in the anterior cingulate cortex with a gradient. So they are really associated with that limbic sensory function in the frontoinsular cortex and limbic motor function in the anterior cingulate cortex. So altogether, this is a system that regulates your general sense of homeostasis. Tells you how you feel, and is involved also in sense of empathy, which permits you to read the state of mind of other people, and after that, get general judgment on social contexts and situations. So that's an important thing to remember. So at first we thought, oh, this is cool. We have a human-specific cell type. So we looked in great apes, and we find them in great apes. So we say now, okay, we have a hominin-specific cell types. And and we studied that with John Ullman at Caltech in a series of great apes, and also across the development. These cells are very numerous about 8 months after birth, and then their number decrease. So there is a peak in the development probably of these autonomic systems. And something quite interesting here, that's a little bit of a vignette. I don't know exactly what it means, but we had one gorilla brain here that had, adult gorilla, that had a lot of cells of these cells in the anterior cingulate cortex. This gorilla is Michael. Michael was the companion of Coco, the gorilla who had learned American Sign Language. And Michael was pretty good at it, okay? And he had tons of these cells. So this is a very special case because it was a human-reared animal with a social environment that was not the norm for a great ape in captivity. So whether this is related or not, we don't know. We really want to look at Coco's brain. Coco passed away July 1st this year. We have the brain, so we'll know. So we did some comparative studies of these cells in great apes. Turns out that humans, as the Economo neurons in humans are much larger if you calculate each of these cells' volumes relative to the brain size. So there is a scaling relationship that does not exist for other type of excitatory neurons. And with Bill Seeley, we looked at what they might express. So first, Bill reported a series of transcription factors like FESF2 and SATB2, a few other things like this. Neuromedin B, which is involved in regulation of appetite in the hypothalamus. But none of this was very satisfying in terms, because it was not entirely clear. More recently, the group of Bill Seeley at UCSF reported the presence of vesicular monoamine transporter 2, very specifically in fork cells and Economo neuron, as well as the theta subunit of the GABA receptors and an adrenergic receptor. So they may be involved in some aspect of or somewhat unconventional modulation of monoaminergic systems in these cortical areas, these particular fields. but the expression at the transcript level and at the protein level is absolutely uncontroversial. It essentially maps that system with these markers. So we start to have a better picture of what it is. Now, what might they be doing? So very interestingly, Bill Seeley found that these cells are exquisitely vulnerable in frontotemporal dementia. We had originally reported that there was a loss of spindle cells in Alzheimer's disease, but the cases we studied at the time were terminal Alzheimer's, very severe Alzheimer's cases. So it's not so remarkable. And in fact, the early stages of Alzheimer's disease do not show a loss of these cells. But if you take the behavioral variant of frontotemporal dementia, these cells form peak bodies, accumulate a lot of TPD43 protein, and are disappearing. So that's very interesting because the behavioral variant of frontotemporal dementia is characterized by a loss of social judgment, social understanding, inability to empathize. And now you have that cell that is probably intimately related to homeostatic function that is primarily affected in that particular type of disease. And that's the behavioral variant. If you take semantic dementia among the spectrum of frontotemporal dementias or progressive non-fluent aphasia, you do not find that effect. It's really only in the behavioral variant. So we did quantitative analysis of that, and it's really specific to the Economo neuron. I'll pass on that. And we looked at other cases where there is a known pathology in the cingulate area. And we looked, for example, at people with agenesis of the corpus callosum. So they developed something called alexithymia, which is the inability to express one's own emotional state. So again, a defect in what we call theory of mind. They cannot read the emotional states of others, and they have no social awareness. These are people who are socially completely disconnected. And what we saw in these cells, in these cases, they are rare cases, is a loss of Economo neuron in the frontoinsular cortex and the anterior cingulate cortex, and a loss that followed the severity of the agenesis of the corpus callosum, implying again that connectivity through the callosum for these cells probably is very important. And the other thing that was quite interesting is that in a case of partial agenesis of the corpus callosum, we saw Economo neurons in an area of the cortex where you normally do not find them, implying probably a difference in the developmental program of the cell, that they go to a place where they have no reason to be in the adult. We did similar observation in autism patients. And we also there see differences in number and densities of Economo neuron in the frontoinsular cortex and the ACC, as well as changes in the ratio of Economo neuron to local pyramidal cells, plus differences in their distribution. And here too in autism, you have an invasion of the orbitofrontal plane cortex by Economo neuron, where normally you do not find them. So again, a major disruption of these cells. We also saw differences in their morphology. They do not look like nice spindles. They get squat. They have contorted dendrites. So really a pathology related to that cell type. And very interestingly, we had the opportunity to study 3 brains from a very rare disease. It's familial dysautonomia, known also as Riley-Day syndrome. And these children are characterized by very severe syndrome that affects all aspects of their autonomic regulation. They have postural hypertension, they have reflux, they can have hypotension as well, they have autism-like behavior, but all of the other function related to the autonomic nervous system are affected. For example, if they are not happy and they cry without tears because all that part simply doesn't work. And it is known it's related to a gene. It affects a protein that is very important for the stabilization of microtubule, as well as the migration and differentiation of cortical neurons. And it's very interesting to see that again, it targets these neurons in that disorder with differences similar to what we had observed in autistic brain. And again, as you can see, with that abnormal distribution in other parts of the brains where they are not supposed to be normally, as if there was an attempt to correct the deficit in the cingulate and frontoinsular cortices. Oh. Yes. So we studied the distribution of an Economo neuron in whales because we kept saying, so is this really just primates? And we went out to whales. And this is, again, the humpback whale. And there my student Camilla Butti did a very careful survey of the entire cortex of the humpback whale. And she found them primarily distributed in the exact same cortical domain, so homology in distribution. And then she extended that study to other whale species like the minke whale, the grampus, the beluga whale, and the pilot whale. And depending on the species, you have more or less of these neurons. The humpback is the species that has the most. Some species don't have much, like the Delphinapterus or the bottlenose dolphin, for that matter. But they are represented among cetaceans. And so that gave us an insight that maybe, hey, in species that are more social or more gregarious or are claimed to have some sort of culture, these cells do exist in larger numbers. Just recently, we had the opportunity to look at one of these guys. This is the bowhead whale. We obtained that from Barrow in Alaska. And indeed, they have these fork cells and von Economo neurons. But there is a very interesting difference in the sense that, as I just say, in the humpback whale and in other cetaceans, they are located in the same cortical domains as where you find them in humans. In the bowhead whales, which is a primitive mysticete compared to the humpback whale, you find them in the entire cortical mantle. That's interesting. Keep that in mind. So, major difference in an animal that is slightly more ancestral with respect to modern cetacean. So we looked at the elephant. So here it's a little bit the same type of thing. You have to get the elephant brain. So it takes going to the savannah in Zimbabwe when there is a government culling of elephant herds, and then you can get a specimen. Paul Menger in South Africa managed to perfuse the brain in situ and got some remarkable histology. Here you have the brain of the elephant. And we looked at that. And in the elephant, it's like in the human, frontoinsular cortex, anterior cingulate cortex, by and large. And we studied the brain of the hippopotamus. And here, you look at the map. It's all over the place. Now, the hippopotamus probably the closest thing to a bowhead whale in phylogeny. So the bowhead whale, which is a balenid, not a balenopterid, a balenid, has retained an ancestral state among these cetacean, okay? So that starts to make some interesting sense. And we expanded these studies to a lot more orders. We find them in some large carnivores like seals and the walrus. So, in horses and zebras. And there again, horses and zebras, like other terrestrial artiodactyls, you find phoneconomo neuron in the entire cortex. So that's something that is a little bit difficult to interpret in terms of the meaning across the phylogeny. Why would certain species have phoneconomo neuron in the entire cortex, and then some species have a much more restricted distribution? So it's as if in some crown species, in certain mammalian radiation, like Afrotherians with the elephant, Cetaceans with balenopterids and odontocetes, and primates, you have a sculpted down, a much more refined type of projection that during evolution in these groups focused on the autonomic nervous system. In these other species, these cells are in a position to have a function like a general cortico-reticular or corticopontine neuron. We don't know. So we are maintaining these collections. They are there to be used. We're trying to expand them with various colleagues. Chet Sherwood at George Washington University. There is an emphasis on primates and marine mammals, but not only. We have about 800 specimens between the two sites that are available, and we are working with a broad network of people in the entire world now, but particularly with Paul Menger in South Africa. We have some interesting data sets with diffusion-weighted imaging for white matter tractography in some of these specimens. That's the brain of Senkwekwe, which you have the picture that was published in National Geographic here when it was poached in the Virunga National Refuge. And we obtained the brain of that animal out of the national park of the Virunga volcanoes, and we have a permanent repository of it. It just remained to acknowledge many people who have contributed enormously to all of these studies, and again, in particular, Peter Morgan and Ilya Glaser. So thank you very much for your attention.